So hi everyone, bonjour, and welcome to our July edition of the CanArt Research Forum's Major Psychiatric Disorders Through a Neurodevelopmental Lens, Implications for Research and Clinical Practice. My name is Eliana, and I'm the Training Program Manager at the Canadian Neurodevelopmental Research Training Platform. Bienvenue à l'édition du mois de juillet des forums de recherche de CanFran intitulée Les troubles psychiatriques majeurs dans une perspective neurodéveloppementale, implications pour la recherche et la pratique clinique. Je m'appelle Eliana et je suis responsable de la formation à la plateforme canadienne de formation en recherche neurodéveloppementale. For those of you who may be less familiar with us, CanArt is a pan-Canadian training hub supporting research excellence and early career researchers through learning, mentorship, and collaboration. CanFran est un pôle de formation pan-canadien qui soutient l'excellence en recherche et les parcours des chercheurs en début de carrière grâce à l'apprentissage, au mentorat et à la collaboration. Today's research forum is presented in collaboration with the Transforming Autism Care Consortium, or what we like to call TAC, a Quebec-based research network that brings together all individuals interested in research on autism and neurodevelopmental conditions. We acknowledge that both CanArt and TAC headquarters are located on the traditional territories of the Haudenosaunee and Anishinaabe nations, a long-standing place of gathering and exchange. Ce forum de recherche est présenté en collaboration avec le Réseau pour transformer les soins en autisme, le RTSA, réseau québécois qui rassemble toute personne intéressée par la recherche en autisme et en conditions neurodéveloppementales. Nous reconnaissons que les sièges sociaux de CanFran et du RTSA sont situés sur les territoires traditionnels des nations Haudenosaunee et Anishinaabe, lieu de rencontre de nombreuses nations autochtones. Today's presentation will be held in English. A recording with French transcription will be made available shortly after. Veuillez noter que la présentation sera en anglais. Un enregistrement avec transcription en français sera disponible sous peu. I will now hand over the mic to today's moderator, Kelly D'Souza, a recent master's graduate of the Educational Psychology program at McGill University and TAC trainee co-lead in training, who will introduce to you our lovely guest speaker. J'ai maintenant le plaisir de céder la parole à Kelly D'Souza, récente diplômée à la maîtrise du programme de psychologie de l'éducation de l'Université McGill et récipiendaire du prix de co-leadership étudiant du RTSA en formation, qui vous présentera notre conférencier invité. Merci beaucoup.
Thank you, Ellie. Dr. Pierre Marquet holds a Diploma of Physical Engineering from EPFL and MD PhD in Neurophotonics from University of Lausanne. Recognized worldwide for his pioneering work on digital holographic microscopy, he is a professor of Université Laval and laureate of the Canada Excellence Research Chair in Neurophotonics. He is also the director of the International Joint Unit in the Neurodevelopment and Child Psychiatry. The goal of this IGU is to define infantile adverse syndrome of major psychiatric disorders such as schizophrenia, bipolar disorder, and major depression disorder by the identification of high-risk biomarkers and endophenotypes with the help of neurophotonic tools. He is the author and co-author of more than 170 scientific publications of many patents and is the co-founder of 3 technological startup companies. It is my honor to introduce Dr. Pierre Marquet and to have him here at our presentation today. Thank you.
So, thank you very much for this nice introduction. Can you hear me correctly?
Yes, thank you.
Okay, great. So it's a pleasure to be here and to provide this lecture. So I will start with the title of my lecture: Autism and Major Psychiatric Disorders through a Neurodevelopmental Lens: Implication for Research and Clinical Practice. The current situation today with autism, schizophrenia, bipolar disorder, and major recurrent depression collectively affect up to 10% of the population. Here, I take the opportunity to make a precision concerning the major recurrent depression. It's really a short part of the depressive disorder. It's what we call long times ago the endogenous depression, which can affect some specific families. So it's completely different from the common depression or the acute depressive episodes that we can have. It's really focused on this kind of endogenous depression with a familial risk. Their clinical expression emerge at different developmental windows from early life to adolescence and early adulthood. Diagnosis often occur late after overt signs and symptoms of functional impairment have emerged. The comments that I can perform here is the fact that today in psychiatry, the only way to perform a diagnosis is to detect this sign and symptom as we perform more than 100 years ago. So it's very subjective and very difficult. And in addition, the presence of this specific sign and symptom which allow us to perform diagnosis are quite late in the framework of the neurodevelopment. Earlier identification may enable timely support and personalized intervention aimed at mitigating the severity of disabling symptoms. This kind of things has been well demonstrated in the context of the early detection of psychosis. And the situation, this specific situation at the psychiatric discipline largely reflects the lack of reliable biomarkers. Why psychiatry is in such a unique situation? Because it's not possible to perform brain biopsies, because really the definitive way to have the right diagnosis of the disorder, it's to perform a biopsy and an analysis, a pathophysiological or pathological analysis of the biopsies. And in the context of psychiatry, neuroimaging currents, even neuroimaging has presented a dramatic progress in the last decades. Currently, neuroimaging lacks the ability to identify specific early pathophysiological mechanisms or even signs and symptoms. And the complexity of the brain, of course, it's contribute to these difficulties. Okay, I have now to introduce some concept or some basic concept around the genetic vulnerability of autism and the specific disorders. So, genetics vulnerability is real, but not deterministic. Familial aggregation is common in autism, schizophrenia, bipolar disorder, and severe recurrent depression, but inheritance is complex. We have incomplex penetrance, means that genetic risk variant increase vulnerability but does not determine the outcome. We have sporadic cases and broad clinical spectra. The key message is the fact that risk can run in families but does not follow simple rules. Complex and heterogeneous genetic architecture, we have common variants. For example, CNP, it's single nucleotide polymorphism. It's frequent DNA variation, each with a small individual effect. We have also rare variants, CNV, copy numbers variation. It could be deletions or duplications of DNA segments, sometimes with stronger effects in comparison with common variants. And we have also the contribution of de novo mutations, these new mutations occurring before birth, not inherited from parents. Somehow, high impact variants can contribute to syndromic forms. Syndromic forms are very severe forms for which there is really no diagnosis issues. But in most common cases, genetic vulnerability arises from the combined effect of many variants, sometimes including de novo mutations. Now we have also a convergence on developmental and synaptic pathways, in the sense that in most non-syndromic forms, there is no single disorder-specific causal gene. Genetics risk is distributed across many common and rare variants with cumulative and interactive effect, as I have just explained before. And these variants can often converge on pathways involved in brain development, synaptic transmission, neurotransmission, plasticity, connectivity, etc. And this means that genetic vulnerability shapes altered neurodevelopmental trajectory in synergy with the environmental factor. Because genetic vulnerability is not enough to shape by itself the neurodevelopmental trajectory, but it's really a combination or a synergetic combination between this genetic vulnerability and environmental factors. So here's a way to summarize this kind of things that I have described. We have this genetic heterogeneity with SNP, CNV mutations, and there is a convergence of this effect of neurodevelopmental process, migration, connectivity, excitability, and so on. And after that, with the combination of this genetic background with environmental, in particular, stressful event and the timing, I will also explain a little bit more about the timing. We have a kind of development of a divergence through the characterization of specific neurodevelopmental trajectory resulting from this combination between genetics and environmental factor to create this heterogeneity of clinical phenotypes or clinical spectrum or spectra, as we said. So we have really a genotypes, phenotypes relationship are many to manys. Okay, now just a few words concerning the developmental components that I have already mentioned. It's had been in particular well documented for schizophrenia, but it's quite the same for others, disorder or autism. It has been demonstrated that before the apparition or the occurrence of the debilitating symptomatology, we have already some alteration in the brain development. It could be, for example, as is described on this scheme, delayed in the myelinization, which is important for the large-scale connectivity, or it could be also a delay in maturation of the inhibitory system or in the excitatory system, which are very important. The balance between them are very important, which has been described. But these discrete alterations are present before the clinical symptomatology. So, if we can to summarize what I have just described is the fact that we have a genetic vulnerability, and in association with stressful life event, we can have the definition or the occurrence of a specific trajectory which can conduct to the pathology or the disorder that we can describe as schizophrenia, depression, major depression disorder, or recurrent depression disorder, bipolar disorder. This stressful event could not only be psychological stressful event, it could be related with infectious disorder and so on. So it could be very large. And for a point important to describe is the fact that the clinical symptomatology becomes specific when we have these famous signs and symptoms that I have described before and that we use with the DSM diagnosis manual. The apparition of these signs and symptoms are very late. And during the neurodevelopment or during this neurodevelopmental trajectory, we don't have any specific signs and symptoms which make the early diagnosis very difficult. And we have the same things for autism, in the sense that the timing is completely different in comparison with schizophrenia or bipolar disorder, in the sense that this stressful event could already be present in a perinatal way or a prenatal way. And of course, the presence of signs and symptoms, the temporality is also completely different, but the concept is quite the same. Now, just a few important concepts concerning the brain development, because I just pick up some important things in relation this neurodevelopmental development of psychiatric disorder. So we have different steps, different processes: neurogenesis, neuronal migration, gliogenesis, symptomatogenesis, etc. But the main point is the fact that at birth we have a quite well developed brain. I would say not completely, not fully developed for sure, but quite well, but not at the level of the connection or the connectivity. For example, per neurons, we have approximately at birth 500 connection per neuron, and in adulthood, you have in an adult brain around 10,000 connection per neuron. So it's a major differences, and these connections are able to be constituted, to be developed during the neurodevelopment only if you have an environmental stimuli. That's a mandatory conditions. So you have the genetics for sure, but you have to have the environmental stimulation to be able to create, to shape this connectivity, this brain connectivity. So there are multiple form or nervous system plasticity associated with the development of this connectivity. You have neuronal plasticity, which is associated with the production of neuron. We have a huge production of neuron during the fetal development. By the way, we have also an adult neurogenesis in the sense that during the whole life, at the level of the brain, there are some specific location in which we have the production of neuron is quite limited, but we have a production of neuron which can be also stimulated by the environment, but it's quite limited. The synaptic plasticity is something which is really related to the creation or the modification of the connectivity, the connection associated with learning and memory processes. Here you have, for example, a strongly activated synapse can undergo structural and functional remodeling to become more efficient when it's well documented currently, and several mechanisms has been identified. We have also a plasticity of brain areas. This is here an example for the pianist. You have it's a neuroimaging neuroimage representation, which shows a neuronal activation in specific areas. So we have for example, primary motor and premotor cortex, which shown this hyperactivation in relation with development of the connectivity. This primary motor and premotor cortex are in relation with fine motor representation of the finger, and we have also primary somatory sensory cortex activation in relation with tactile and proprioceptive integration of the finger. So we have this, the brain has capacity to also show some remodeling of not only a remodeling at the synaptic level, but also a remodeling at the level of specific area dedicated to specific tasks. So the brain wears out if you don't use it. Brains, in particular the cortex, aims at reconstructing an appropriate internal representation of the external world, is something which is in particular important in the context of psychiatric disorder and autism. It's an important task of the cortex to be able to process correctly the information coming from the vision, audition, olfaction, taste, flavor, and also from the antero and proprioception. And to do that, we have the sensory information coming from the different primary sense should be processed, but also we have the brain have to perform a multisensory to have this appropriate internal representation of the external world. And associated with this multisensory integration, we have the development of emotional aspect, and we have affective response associated with this internal representation of the external world. This affective response become conscious at the level of the prefrontal cortex, and in this information is processed in a cognitive framing, leading to a specific action and behaviors. So this challenge for the cortex to perform an appropriate internal representation of an external world is sometimes referred as the binding problems, how the brain links different types of information, multimodal world, to a form of coherent whole and keep tracks of it in memory. So, nervous system to perform this kind of task faces a constant challenge how to maintain flexibility and stability at the same times. So we have described the adaptation to new situation, learning and memory associated with the local plasticity, in particular the synaptic plasticity. But there are also other mechanisms to maintain the firing neuronal capability within a sensitive working range, the optimal state for input processing, which is called the homeostatic plasticity. So, in a few words, homeostatic plasticity, neuronal circuits do not only change, they also stabilize themselves. Homeostatic plasticity helps maintain neuronal activity within a functional range, and a key mechanism is a global multiplicative synaptic scaling. Neurons adjust synaptic strength up or down proportionally across their synaptic input. In fact, at the network level, the stabilization also involves the regulation, and it is an important point, a regulation of the excitatory and inhibitory activity. The excitation and inhibitory balance is therefore a key readout of this network stabilities. So monitoring the excitatory inhibitory balance dynamics may help assess neural network vulnerability of resilience. Now, another, it's the last concept that I want to introduce here because it's really important one within the framework is critical periods. What is a critical or sensitive windows in cortical maturations? Brain development is organized around multiple times windows or increased plasticity and vulnerability. These windows can occur at different times across the brain, across region of the cortex that support different functional system. And they contribute to the maturation, this different critical period of sensory, motor, emotional, cognitive, and social function. And during this period, synaptic and circuit plasticity are high, allowing a rapid adaptation to experience. It's an important point. The opening and closure, because each critical period has a beginning and an end, are shaped by the excitatory inhibitory balance. Synaptic pruning is also present during this critical period, inhibitory maturation, and also perineural nets. So the main message here is that there exist at the level of the cortex different critical period of very high sensitive windows, which are both windows of opportunity and windows of vulnerability. So here I indicate some important critical period associated with the development of the cortex. And with different temporality, you have, for example, early sensory critical period, processing of sensory information, and so on. But what I can mention from this table is the fact that, for example, for schizophrenia, bipolar disorder, and severe recurrent depression, adolescence is probably not when everything starts, but rather when earlier subtle developmental alteration become clinically visible, associated with the critical period of the prefrontal cortex and limbic circuits, which undergoes pruning, reorganization, and late maturation. So it's the reason why, probably why this disorder emerged during the early adulthood, adolescence, during the adolescencehood, in association with the critical period of the prefrontal cortex in relation with social and emotional maturation. We have a similar logical for autism, but autism is probably more associated with the critical period associated with early sensory system development. So it means that in these schemes that I have already presented before, the main message associated with the critical period is the fact that the stressful event, the timing from the stressful event is very important to in a sense that if a stressful event occurred during a specific critical period or not, the effect could be completely different and the neurodevelopmental trajectory could be completely or drastically modified. So the timing associated with the critical period of the stressful event is very important. It could explain also the large diversity of the phenotype, clinical phenotype. So our research hypothesis around all the things that I have exposed is the fact that altered multisensory integration could be an early developmental vulnerability biomarkers. So our program research to test such an hypothesis involve high-risk cohorts for such biomarker identification in the sense that we have the goal to detect two different types of biomarker, trait biomarker, as is described here in the sense that the trait biomarker could be present before the apparition of the clinical phenotypes. And we have also state biomarker, which are present or associated with the clinical phenotype, which could be, by the way, helpful to perform an early diagnosis. We have the same concept for autism, which is not represented directly on this scheme. So the goal is really to identify trait or neurodevelopmental biomarkers and also states biomarkers, which, as I already explained, could be helpful for early and accurate diagnosis and also to guide timely and personalized therapeutic intervention. In contrast, trait biomarker could be important to monitor the neurodevelopmental at-risk trajectory quantitatively over time and also to provide deeper insight into the early pathogenesis of this major psychiatric disorder or autism. So to perform such a research, we have to work with high-risk cohorts. For example, we have access to study of families at high risk for severe psychosis. It's a study of families in Eastern Quebec consisting of family affected by schizophrenia, mood disorder, or over several generations, which has been recruited by Dr. Maziad at the Cerveau Center. This is a typical sample that we have in such a cohort. But as I also have the chance to lead the joint research unit between the University of Lausanne and Université Laval, I have also access at the Lausanne-Geneva high-risk cohort, which is constituted of bipolar patients, major depressive patients control, and their high-risk offsprings. So what kind of research we can conduct in relation with the multisensory integration? This is different phenotyping platforms that we have in association with this high-risk cohort. So today I will describe some research in association with the multisensory integration, and also some research in the second part of the presentation of the research program in association with neurophotonic platform, something more in relation with cellular aspect of this disorder. So the goal here is really to detect different type of biomarker, trait biomarker, state biomarker, and to put together all these biomarker to be able to have a chance to characterize the neurodevelopmental trajectory or the presence of the disorder to perform early diagnosis. So what we perform concerning it's a clinical research concerning the multisensory integration. We test the intermodal transfer. The intermodal transfer is the ability to transfer a percept from one modality to another. So, for example, a well-developed by 6-year-old of age, it can be tested with 3D shapes in different modality. For example, we have 3D shapes that we perform with 3D printers, and this shape could be quite complex. And the goal here is the visual tactile conditions. Such a shape is presented to the participant. It's Valerie and which performs such a task in the lab, and so she has just presented a specific 3D shape to Martin, and after that they give to Martin this specific shape in a black box, and Martin can handle the shape, the 3D shapes with his hands, and he has to say, okay, this one corresponds to this one, and so on. And it's not very easy. And we do this kind of task. And for example, we have some results in this visual tactiles dimensions that for controls in comparison with offspring of having um, parents suffering for bipolar or schizophrenia here, or for a major depressive, recurrent depressive disorder here. And this is a result, is preliminary result, of course, but between, for offspring between 9 and 15 years old, and we have some slightly difference which are not statistically significative, but when we decipher this result by a different period of age, we have a significant, statistically significant difference between control offspring of bipolar and schizophrenia patients, and also offspring of major depressive disorder, which has difficulties in this visual tactile intermodal transfer. So it means that this high risk offspring, it's of course preliminary result, seems to exhibit some difficulty in this kind of task, reflecting a multisensory integration. We have also started to develop another approach called vestibulography. Why vestibulography in the context of a multisensory integration, as I have just explained before? That's a collaboration with uh, uh, it's not Brian, it's Brian, sorry for that, Brian Lightgo for the University of Winnipeg, which has developed this very accurate and precise technique called eVest. And while we test the vestibular system, to do that, we use a chair which has the capacity to rotate in different direction to stimulate the vestibular system, and we have an electrode introduced in the conduct of the ear to detect the vestibular response associated with this stimulation due to the chair rotation. Why the vestibular system? Because the vestibular system plays a central role in orchestrating multisensory integration across bodily, sensory, and motor signal. By the way, this technique has shown its capacity to detect specifically schizophrenic patients, bipolar patients, and to perform a differentiation between schizophrenic patient, bipolar patient, and also major depressive patients. So it's a very promising techniques to test, to assess the multisensory integration that we have started to use in the lab. Now, we have also another platform associated with what we call the neurophotonics, because what is the neurophotonics? In my lab, we have a long tradition to develop new cellular imaging techniques using optics. That's the signification of the word neurophotonics. So, as I explained before, it's not possible to perform brain biopsy for obvious reasons. So, nevertheless, to be able to obtain some human neurons coming from patients or having the genetics vulnerability of patients. I explained quite a lot now about the genetics vulnerability of offspring and patients. We use a technique which has been developed approximately 2 decades or 1 decade ago, which is the production of iPS cells. What is the iPS cells? In fact, we collect on patients and their high-risk offspring some very accessible peripheral cells, for example, fibroblasts or urothelial cells coming from urine collection. And by performing cellular reprogramming, which is a procedure which has been developing by Shinya Yamanaka, who has been awarded by the Nobel Prize for this discovery, we can obtain what we call some induced pluripotent stem cells. So it's a kind of stem cells which has the capacity to be neurodifferentiated in any kind of cells of our body. So when we have obtained from fibroblasts or urothelial cells this induced pluripotent stem cells, we perform a neurodifferentiation of these cells, first in neural progenitor cells, and after that in neurons and astrocytes. So we can have neuron and astrocytes having the genetics vulnerability of patients and high-risk offspring coming from our high-risk cohorts in a Petri dish. So the goal here is to say, okay, we have the genetics vulnerability of the patients, and we can follow accurately the neurodifferentiation, how the neuronal network is performing connection or is its maturation in the sense of the inhibitory and the excitatory system, as I explained before, the importance in the context of the multisensory integration. And by the way, we can also stress these progenitor neuronal cells in culture by performing some stressful event to mimic, to reveal this genetic vulnerabilities I explained before. So to perform this neuro monitoring or the monitoring of this neurodifferentiation process, we have developed a specific techniques called polychromatic digital holography microscopy. I don't want to go in details here, but what we can do with this technique, it's label-free techniques, just some image that we obtain with this technique to illustrate the capacity of the technique here, is astrocytes in culture, very flat cells, very difficult to observe these cells. And we have the accuracy to observe a very thin part of these cells, which is called a pseudopod or lamellipod, essentially a thickness of two membrane. It's approximately a thickness of around, I would say, 20 or 30 nanometers, and we have this capacity to flow this thin membrane part of the cell moving in the perfusion chamber. So it's just to illustrate the sensitivity. We have also the capacity to see inside the cells without any labeling. So suddenly you will see it's a neuronal culture, a culture of living neurons, and we can have we can visualize inside the cells, the nucleus, and so on. But we have also have the capacity to monitor the connectivity, the development of connectivity. Here is just to illustrate you the sensitivity of the approach. We can observe the fluctuation of red cells membrane because it's well known that red cells exhibit a nanoscale fluctuation, so at the level of the nanometer, and we can observe this nanoscale fluctuation of red blood cells just to illustrate the sensitivity of the method. What we with this method, we extract what we call a quantitative phase signal, which can extract a lot of different things concerning neuronal process, neuronal maturation. But today, I will focus on two things that we can perform with these techniques. Here, we have recorded during several weeks the neuronal maturation. So at the first time of the monitoring, you will see it's a loop, so you see always the same period of time. So at the beginning of the loop, you can observe neuronal cells or trying to make connection, progenitor neuronal cells trying to make connection. And after we have the development of the neuronal network, the connectivity. So we can monitor, for example, neuronal proliferation, neuronal migration, synaptic formation and plasticity, network connectivity, and also the functional maturation. In particular, the inhibitory and the excitatory system. I will describe this part in a few minutes. So something which has been very interesting to extract from this monitoring with the polychromatic digital holographic microscopy is the topology of the network. And we perform this with a close collaboration with theoretical physicists which are experts in what we call graph theory. Graph theory, in a few words, are able to say very important things about the network by extracting nodes and edges. And by doing such an extraction, graph theory can reveal, for example, the resilience of a network, the robustness, or in contrast, the vulnerability of the network. And by doing this, we have started to monitor the fact that when we are dealing with cells coming from schizophrenic patients, the development of this neuronal connectivity is different and present a level of robustness and resilience which is decreased in comparison with control. Now we are trying to test this kind of things of offspring because it could be represents a nice trait biomarker for schizophrenia. That's the extraction of the key concept behind the graph theory, the nodes and the edge. Okay, now an important point that we want to monitor is the excitatory-inhibitory balance, because I explained before why the excitatory-inhibitory balance is important in the context of the homeostatic plasticity and the treatment of information to perform an integration, a multisensory integration task. So how we can measure this kind of excitatory-inhibitory balance is based on the fact that mammalian cells are permeable to water. Neuronal firing induces some ionic movement, and the electrical activity of neurons, which drives osmotic water flux across the membrane for osmotic reasons. And as mammalian cells are permeable to water, we have a chance when water goes inside or outside the cells to change the optical properties of the neuron, in particular the intracellular refractive index. And that's the case as our quantitative phase signal coming from DHM is sensitive to the intracellular refractive index, an optical index, it's an optical signal. So we are able to monitor specifically the activity of ionotropic receptor. In particular, we test the mature neuronal network which exhibit or which has to exhibit a balance between excitatory and inhibitory system. One of the main neurotransmitter in the brain associated with the excitatory system is glutamate. So we test the effect of glutamate on a specific, very important ionotropic glutamate receptor, which is called the NMDA receptor. And this NMDA receptor, when this receptor is opened due to the presence of glutamate, we have an entrance of sodium, which provokes neuronal depolarization associated with an entrance of water, which will modify the intracellular refractive index of our signal. So when we perform this kind of recording, it's again interesting because this NMDA receptor has shown to be altered in schizophrenia. And when we perfuse glutamate on such a culture of living neuron in culture, we have this kind of pulsation. And when we have an accurate look at this pulsation by performing a cross section, we show a decrease of the cross section corresponding to an entrance of water associated with the activity of the NMDA receptor. And after that, we have the neuronal or the volume recovery, because it's important that associated with this water increase, we have a modification of the volume of the neuronal body, which is tightly regulated. Again, it's a loop. So we are able to monitor with this technique the level of activity of this specific NMDA receptor associated with the excitatory system during the neurodevelopment. And we do the same for the GABA system. The GABA system is really the main inhibitory system. And we perform the same monitoring of the GABA system by perfusing GABA during the neurodevelopment, during the neurodifferentiation of this neural progenitor, neural progenitor cell. And for the GABA, it's a little bit complicated because when a neural network is immature, GABA, in fact, has an excitatory effect, a paradoxal excitatory effect, due to the fact that when you open the GABA receptor, which is a chloride, ionotropic chloride receptor, when you open this GABA receptor, if the level of chloride inside the cell is quite high, you have an exit of chloride, so you have a depolarization. And the level of chloride is quite high when you have an immature neuron. And when you have a mature neuron, due to the presence of specific co-transporter, which are indicated here, the level of chloride or the concentration of chloride become quite low. And when you have the opening of this receptor due to the presence of GABA, this GABA receptor, you have an entrance of chloride, so an entrance of water. So if the GABA system is immature, we have an exit of water. If the GABA system is mature, we have we have an entrance of water. So we test these kind of things during the neurodevelopment. And again, when the neuronal network is quite young, we have an increase of our signal, which reflect an exit of water. And when we have the translation, which is called the GABA switch, to go toward a mature GABA system, we have an entrance of water, so a decrease of the signal. This research is particularly important in the context of autism, because we know it has been described a delay of the maturation of the inhibitory system. So we have started to develop this project in the context of autism, in the sense that we have collected some urine cells coming from patients having autism, or for newborns at risk of having autism, to test this inhibitory excitatory balance. So by performing this same protocol that I described, but directly at the level of fibroblasts, without performing all the reprogrammation to obtain neuron, we have started to detect a biomarker, a cellular biomarker for bipolar disorder. Here it's a collaboration between my lab in Lausanne and my lab in Quebec. We can perform a multi-center studies, which is important to detect or to identify a biomarker. And here for control, we have a specific level of this fibroblast. That's the fibroblast response to stressful events. And from fibroblasts of control and major recurrent depressive disorder, we have no difference. But for patients having bipolar disorder, we have a clear difference. So their fibroblasts, their response, their fibroblastic response to stressful event is very different. And this is a very promising biomarker because the specificity is quite high. So now what we can do with these cells, we try to detect some specific phenotype, as I explained before, coming from patients-derived cells for which we perform a reprogrammation and differentiation in neuron cells. We are trying to detect some specific phenotype on our biomarker with all the process that I can mention just here to perform image processing and so on. And when we have a disease-specific phenotype identification as for the bipolar disorder, after that we can test some specific drugs if you are able to normalize this disease-specific phenotype. And these drugs could be in different conditions. It could be drug response prediction to perform a personalized medicine, because in psychiatry it's very difficult not only to have the right diagnostic, but to find the right medication, but also to perform drug repositioning and to identify new druggable targets. So by having presenting all these kind of things, what I can say, if I try to summarize, if we are able to detect such a biomarker, a risk biomarker or risk endophenotype, which are illustrated here by these different pieces, we could be able to, in fact, to reduce the heterogeneity of the clinical spectra through the identification of subtypes based on the presence of such biomarker or endophenotype, which also could perform a reassessment of psychiatric nosology, the description of the disorders that takes biological component into account, because today, as I explained before, we don't have any reliable biomarkers. So in the psychiatric nosology, we have absolutely no biological component to classify the disorder. And these subtypes, having a component, a biological component, would include via biomarker and identify biological component, leading to a better understanding of the pathophysiology, because we don't know very accurately today the pathophysiology of autism or of this major psychiatric disorder. And when you have a biomarker, you can think in terms of personalized medicines, and also risk biomarker. Infant risk could help to define infant risk syndrome, to be able or to start to think in terms of informing prevention strategy. And also stronger biological anchoring of psychiatric disorder and autism. It's very important to go toward a destigmatization of these difficulties. And this could be, if we were able to detect this kind of biomarker of endophenotype, it could be really a paradigm shift for psychiatry in this developmental scenario. So this work has been or is currently performing by my team in Quebec at Cerveau Center, and also with my lab in Lausanne, associated with the CHUV, the Centre Hospitalier Universitaire Vaudois. And we have plenty of collaboration, for example, with the Center for Psychiatric Epidemiology and so on, to have access to this high-risk cohort. So thank you very much for your attention. It will be a pleasure for me to answer your questions.